Height for you, madam. That is height for you.
अच्छा ये पिलांकर आरंभ कर दो तुम सिद्धांत हाँ आरंभ कर दो सुधाकर सर कहते हम आरंभ कर दो हाँ 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 बोल
ठीक है
हाँ ମୁଁ କହିଛି ପିଲାଙ୍କର ଆରମ୍ଭ କରିଦଉଛି ମୁଁ ତମେ ଆସ
ठीक है तो ठीक था उससे कंटिन्यू करो
ये ठीक है
Essential behaviors. So these are the cells which survive post-therapeutic stress, including pivotal therapeutic resistance and deep neural relapse. So now we want to basically a lifestyle-based degradation process in which unnecessary environment proteins are involved in the development of structure from pharmacogens, which finally lead to degradation of the proteins. So these are the whole process in the autophagic process. And individuals use to study the effect of chloroquine. So basically, this study we use to their effect on cancer cells. Basically, like aspects of cancer, metabolism, migration, stemness, and evolution. During the this of we basically deal with the stemness and evolution. Now coming to reactive oxygen and antigen. So NRF2 is basically the key constitution factor that we do the its relation to the loss in hospital. One is active, that means other is less level. So basically, high NRF2 leads to sensor level and proliferation. So to better understand, so to better understand the mechanism, like how cancer cells basically escape, we need to understand the connection between autophagy and antigen with oral cancers. Now moving to the result section. So we basically use two techniques basically steroid acids as well as max assisted cells of this to separate cancer cells from cancer cells. So what we have found that in cancer cells, the stimulus markers are significantly high along with the low expression of apoptosis markers, suggesting that these cancer cells are apoptosis resistant. Now we expose these cancer cells to about 6 and find out the apoptosis rate is significantly low in cancer cells than their CD40 minus counterpart, suggesting that cisplatin treatment induces apoptosis resistant CD40 positive cancer cells in oral cancer cells. So as I already mentioned that autophagy plays a major role in recruiting the cancer cell death and chemo resistance. So we then we evaluate the autophagy status and we found that when these cancer cells are exposed to of the period for different time points, autophagy marker holding that patient consistency. Next, so what we as lifestyle plays a key role in regulating autophagy. So we measure the lifestyle activity using different experiments. So what we found in systematic cells, these CD44 positive cells for higher lifestyle activity. And here we use peptide that is basically cathepsin D whose expression is high. We found that in presence of peptide A, the active lysosome, that is basically denoted by lysosome rate, is significantly abnormal, suggesting that lysosome activity plays a major role in regulating for lysosome activity to response chemotherapy stress. Next, we measured the autophagic flux, that is basically the whole degradation part, and found that in presence of these two units, chloroquine peptide there, the autophagic flux was inhibited. Next, as I already saw that 85 rating of the expression was significantly high in cancer cells. We logged down these two proteins using SH85 and SH50. Now we found that in the absence of these two genes, autophagy level are significantly low in cancer stem cells. Then we are learned that their effect in cancer stem cells, and we found that these in the inhibition of these two genes, cancer stem cells is reduced. So suggesting that ATC and nuclear dependent autophagy are crucial for stem cells and protection against depending on the approach. Then we use the mitochondrial loss, like how the loss plays a role. We found that CD40 positive cells when exposed to cisplatin, there was higher amount of loss, and we found that when we autophagy was inhibited, in that case the amount of mitochondrial loss was high, but should not exceed the CD40 minus cells, suggesting that any alternate mechanism might be played in the absence of autophagy. So what we do, then we targeted the NRF2 signaling and found that in the absence of NRF2, the mitochondrial loss increase as well as cell viability is also reduced, suggesting that NRF2 controls mitochondrial loss level in autophagy cell also exposed to cisplatin. Then we come to physiological variables. We found that in the absence of these both clonopin as LPA5, the complex NRF2, significantly reduce the cancer stem cells as well as increase the apoptosis in CD40 positive cells, suggesting that these NRF2 signaling agents used into therapeutic efficacy of cisplatin. Now coming to the conclusion, so cancer cells basically are autophagy inhibition to activating NRF2 signals, simultaneously inhibition of both the signals. It improves therapeutic efficacy of cisplatin by inhibiting cancer cells associated drug resistance. Now coming to so I'd like to acknowledge providing the service. Thank you.
Wonderful.
Nice name you have given my daughter. There's a death in the name of the lab.
So, any suggestion from my professors?
Nice presentation. He stuck to the time. That way he should get the great mark.
Yes.
Next presenter will be Sunita.
For a healthy environment, predator and the prey relationship must be maintained.
Please introduce yourself.
In natural condition, the natural enemies have a play a major role in controlling the predator relationship. Here, I am Sunita Dr. working as a research scholar in Biological Survey of India, presenting that how parasitoids play a natural enemy role as a predator as per our that spider as a predator and the parasitoid Hymenoptera as a natural enemy. So spiders are the megadiverse group in our ecosystem and they can eat enormously arthropod population. They can eat everything. More than 50,000 species are reported globally. So according to the World Spider Catalog, India has 1,907 species under 490 genera in 61 families. While Odisha have 267 species under 162 genera in 42 families. So here we can count that how diverse the group is. So before coming to, I wanted to know how and what are the natural enemies. 4 different kinds of natural enemies are there. One is Hymenoptera, other is Diptera, and Lepidoptera and Neuroptera. So these are the families in which they are act as a natural enemy which are attacking spiders. So 3 broad categories of natural enemies are there. One is type 1, another is type 2, and another is type 3. Type 1 natural enemies are those enemies who lay eggs on the host egg. Basically, they are solitary, means single egg, they are taking their nutrition to develop further. That's why they are called as type 1 natural enemies. Type 2 natural enemies are taking their nutrition from the cluster of the egg mass. And type 3 are the ectoparasitoids who develop and depend upon the spiderlings and the mature spider for their nutrition. So in this article, I am going to tell only the type 1 classification, type 1 natural enemies who are attacking the spider with a case study. So if you consider the Hymenoptera, that is the third largest order in the insect order. 75% of the Hymenoptera are parasitic in nature. In that, Celionidae are the exclusively egg parasitoid. Means all the Celionids lay their egg in the host egg itself and develop the further development. So in that Celionidae, total 10 genera, 10 tribes are there. Depending upon the host they are taking. So under the 10, tribe A is the only tribe who takes spider as their host. So in world, total 8 genera are present in the tribe A, but in India only 4 genera are there. So these are some examples of type 2 natural enemies. I am not going to elaborate that because time is very short here. So in the case study, so meet our host spider. These are the clubunid spiders. These are the commonly known as sac spiders. All the spiders do not make web. Some make retreats and some make web. Clubunids are the sac spider because they make a retreats in the leaves, in the rolled leaves, with both ends are open. So and they lay their eggs inside the retreats, and also they mate inside the retreats itself. So, and basically they are nocturnal and hunter spiders are present. So globally 639 species under 15 genera are present. In India, 35 species under 4 genera according to the World Spider Catalog 2022 is concerned. Clubunids are basically attacked. Since I am taking as a group which are exclusively egg parasitoid, means all the group of the members lay their egg inside the spider eggs. In that, only Ceratobius and the Bias are the 2 genera are reported earlier that clubunid spiders are their host. Till now, only one species of the clubunid, that is Clubiona dracops, reported from the eastern part of the Moykanj, Odisha. Means you can know about spider population as well as the parasitoid population are less explored in Odisha state. So, these are the only literature found in our India. There is no much. So that only India 2 host reports are there. Not much are worked out. My study area is the sample collection was done in the 10 agroclimatic zone. More than 50 samples are converted. The case studies are reporting that the spider collection and population from the Bamara block of the Sambalpur district of the Gandharva region forest, which is majorly sal forest. From there, the egg was collected, and from that egg, the parasitoid emerged. That is our heroine of the story because Hymenoptera is the group in which female plays a major role rather than male play a major role. So really it was done by the normal that general method. I am not going into detailed methodology. So instruments are used for identification and photography. So parasitism rate was the Sarietal that 2021 the recent method, and molecular characterization with the ask for the already told. So the result from the one clutch, 44 eggs are there in which 22 spiders and 22 Hymenoptera parasitoid emerged. And the interesting thing was that 22 are the female. No males are emerged from the spider ring. No spider ring emerged. That can you understand the rate of parasitism in the spider, that parasitism rate was 50%. And this is the species which is the reported the host as the Clubiona filica. This is the first report from the state. None other species of the Clubiona filica was reported earlier from the state. That both that morphological and the molecular will be attended. That emerged parasitoids will be having tridentated mandible, compact clava, full hind wing with complete submarginal brain, and that third metasomal turbulate with. So, and this is the new species what is emerged from from the spider egg. So this that due to this character, this is called as genus belong to the Ipris. So as per the taxonomic documentation followed by Rajmona 2014, this is the most close related to the species Ipris tuberensis. But due to that marked area, that the absence of notule and the absence of striation in the third tergite metasomal tergite, it is clearly understand that this is a new species, not a earlier reported one. Significance of the study, as we know, these are the population who are controlling the natural enemies of the spiders, so they play a major role in balancing the ecosystem. So summary of my work: new host and parasitoid association was reported first time. First time, both the host and parasitoid identified up to species level from India. None of the species was identified from the India. Only family level identification was done. First record of the host species from the state, and novel sequence of the generated. None of the every species sequence have been generated before from India. This is the first time sequence generated from the genus. These are the some of the reference, and I am very much thankful to my director, ZSI Zoological Survey of India, and Minister Environment, Forest, Climate Change for giving me the opportunity and the placement and the funding. Thank you.
If any question or suggestion, only one.
I would say nicely presented, very cogent to the focus and to complete within the prescribed time.
Very good presentation. Thank you. Thank you very much.
Thank you, sir. Thank you for your patience for hearing a good thing.
Next is Ashish Kumar Sahu, followed by Vikram Kumar Senapati. Ashish now.
Respected teachers, member of RABS 2022, myself Ashish Kumar Sahu, standing before you to keep my topic. Yesterday, whatever lecture we have heard, we have known only one side of the story. We are now entering to hear the other side. Yesterday, sir, we were discussing about rice cultivation, its diversity, but now let's once after green revolution meant that we use pesticide. So after using pesticide, what are the effects that farmers face? Let's hear. So we all know what is pesticide. Pesticides are the substances that are intended for preventing, destroying, repelling, or mitigating pest. So what are the problems that we face when we use pesticide? First thing is that pesticides are persistent. Second, though it is persistent, so it stays in the environment for longer life period. And third, though it is persistent and remain in environment for longer period of time, it is biomagnified. That is from one food chain, it transfer from one trophic level to another, and its concentration get increased in the higher trophic level. Then here you can see these are the classification of pesticide. I am not going into the detail most of the time. You can see that by target organism, it can be herbicide, insecticide, fungicide, and by chemical family when we see three category: organochloride, organophosphate, carbamide, and chemical structure where you can divide the organic inorganic. So these are example of some of the pesticide. You value DDT is organochloride. Then second, now here you can see that why pesticide is essential. So when there was feline, to meet the need of feline, definitely food production has to be increased, and to increase the food production, we use that is technology of green revolution, and we use the pesticide. So again, you can recall from yesterday what Sanjay Kumar Sahasar was telling that the energy whatever an organism has, it can divert either into its growth or for its immunity. So here we are only targeting growth by using pesticide. So while we are increasing the growth higher to get higher variety of rice, we are compromising for the immunity, and that's when pest get attack and the plant is less resistance. So So why we are concerning about pesticide? First thing is that pesticides are hemophilic in nature. So they are so they can, as they are hemophilic in nature, so they can cross the blood-brain barrier easily. And so they can cross the blood-brain barrier easily. They can enter the brain, and they, as you know, neural cells are big, non-dividing cell. So whatever the number of neural cells present in the body, that remain constant, rather it get degraded. So neuron cell get once neuron cell get degraded, we are unable to our whole neural system is hijacked. So then you can see that I am not doing in the detail mechanism of cholesterol activity, how it get attached to the cholesterol and unable to regulate the control mechanism. So so this is when got the we found the hypothesis that neural cells are not dividing and they get degraded, they are getting degraded. So what if farmer are exposed to the very stress? So they are unable and they are incapable to cope with the stress. So then we found that so then we thought that prolonged innovation multiple forward is a we do we do farming there for 2 seasons.
What method?
So these are excessive pesticide with local lifespan can lead to their accumulation in the soil, and as it get accumulated in the soil, it has the chances to get transferred into the rice paddy, and whatever we are eating, then we are exposed to pesticide in many ways. By eating by food, we can also farmer those who are spraying the pesticide, they may get directly exposed by the nasal route into the brain. So these are the here I have shown that what are the farmers who follow the rules when they are spraying the pesticide, but they are not following. So this is my sampling area, Borgar district, where I have chosen as a case study. And after visiting the field, we found that the field is designed in such a way that the irrigation pattern is canal system. So from canal, water is transported from upper land to subsequently middle land and lower land. So this is a review of literature that I have done. And so when I characterize the soil, I have used these parameters. Here I have shown. Okay. So here we can clearly see that when we see the nutrient like phosphate and nitrate, we can clearly see that the nutrient is getting leached from the upper field to middle field and to lower field. And when in S2 wells, here I have seen that there are 24 major peaks of pesticide that are residue getting delivery. So maximum level of the pesticide residue that we found are chlorpyrifos and chlorpyrifos pesticide. This is the multiplication of pesticide we have done. This is the whatever the red area I have marked that are above the permissible limit. So third page I have said that I have now to study how the farmer are getting affected. In that we have done questionnaire and done certain tests. So this is standard elemental state examination where we study the cognitive function of the farmer. There farmer have to simply draw a line between the dots. If they are properly drawing, then their neuromuscular action is right, and if they are unable, their second test is finger tapping test, where they have to tap the finger with 1 minute 60 seconds. So their score are recorded, and here we are. These are the participants actually I have taken, and we are seeing here we can clearly see that the score of the farmers are decreasing when the farmer are exposed to more number of years and simultaneously more number of hours. So then this is half of my work. Also done more to this is only the problem was discovered and thank you.
Actually, this is a nice piece of work, but this right naturally. Within 5 minutes, it's not possible. If you want to comment it, thank you. Thank you very much. So we have Vikram Kumar Senapati introduce yourself. Will be followed by Sanam Kumari or Sanam Kumar Saul. Sanam Kumar Saul.
Good afternoon all. I am School of Life Sciences, Koraput University, under the guidance of Dr. Vikram. This is my topic. The non-photosynthetic pigments of leaves provide the resistance against the UV radiation in contrasting rice cultivars. As we all know that the chlorofluorocarbon, which is a greenhouse gas, is mostly responsible for depletion of ozone layer and allow the unwanted UV radiation to reach into the Earth's surface and have a harmful effect on both the plants and animals. But in comparison to the animals, the plants are directly exposed to the UV radiation as they are immobile and stationary in nature. The various harmful effects of UV radiation, like the leaf bronzing, chlorosis, inferior grain quality, membrane damage, reactive oxygen species, generation. No doubt, the semi-dwarf high-yielding variety have the higher productivity rate, but unfortunately, they are quite sensitive towards the UV radiation. On other side, the western part of Odisha, which is endowed with a rich diversity of the various traditional and wild rice variety, and these traditional variety are resistant against the various abiotic stress, including the UV stress. These are the some traditional variety like the Lal Koi, Ora, Alavati, and the white rice variety that is Orisa Nivar and Urjivon have developed a various strategy against those UV radiation for their protection against the UV radiation. So the aim of my objective is to identify the particular mechanism of UVB resistance in traditional rice cultivars by the morphophysiological character and also the various analysis like the biochemical, phytochemical and absorbance fluorescence analysis. And finally, the assessment of the UVB induced grain sterility and its impact on the grain. The entire experiment was done in the Botanical Garden of School of Life Sciences, Jeypore University during the session of 2022. The two rice cultivars was taken. One is the Lal Koi, which is a purple traditional variety, and another is the Swarnaprabha, which is a high-yielding variety. The supplemental UV radiation was provided, that is 5 hours per day, which energy content is 14.4 kilojoule per meter square per day. And the distance between the UV tube and the canopy of the plant is always maintained, that is 20 centimeter according to the height of the plant. The treatment was starting in the rooting stage and it is continued up to the 30 days. The sampling during the 10, 20 and 30 days after the treatment and analysis of the various parameters. This is the experimental site where the UV treatment was provided to the rice plant. In morphological analysis, it is clearly revealed that in a large variety after the UV treatment, there is no leaf burning effect is found. Whereas in the Swarnaprabha, it is clearly visible that after the UV treatment, the leaf is become burnt. And A and C is the simple and scanning electron microscopic view of the Lal coin leaf surface, which indicate that it has contains the higher amounts of the pubescent hair in the leaf surface, which is act as a reflectance against the UV radiation. Whereas in case of the B and D, which is the simple and scanning electron microscope view of the high-yielding Swarnaprabha variety, it has very negligible amount of the pubescent hair that indicates their sensitivity towards the UV radiation. After the morphological analysis, we focused on the various phytochemical analysis. The flavonoid is estimated by the concentrated sulfuric acid test, the phenolic compounds by the ferric chloride test, and anthocyanin by the HCl test. And all the phytochemical tests have a positive sign towards the traditional Lanmai variety that it has. They have the high flavonoid content, the rich in phenolic compounds, and have the anthocyanin content rich as compared to the Swarnaprabha variety. After phytochemical analysis, we estimated the various biochemical parameters. The total chlorophyll and carotenoid content is clearly indicated that after the UVB treatment, it has the very high decline in case of the Swarnaprabha than that of the Lal coin variety. In Flavonoid content in all the three wavelengths, that is 270, 300 and the 330, is clearly indicated that UVB resistance activity was found in case of the lal pine variety, and the similar result was obtained in case of the anthocyanin, that it increased their anthocyanin content after the UVB treatment in lal pine variety, whereas the Swarnaprabha it is gradually declined. The lipid peroxidation content was very much high in case of the Swarnaprabha than that of the lal pine variety, that indicated their reactive oxygen species generation. After the biochemical analysis, we focused on the adsorbance fluorescence mechanism. All the two varieties have the higher adsorbance after the UVB treatment, but only the Lalmoy variety, which is a traditional variety, have increased their fluorescence rate and compared to the control that which fell in case of the Swarnaprabha, it is gradually declined their fluorescence rate. And finally, we estimated the percentage of sterility. It is clearly indicated that the Swarnaprabha variety has the sterility that is 9.43, but after the UVB treatment, the sterility is very much high. My conclusion is the non-photosynthetic pigments, basically the anthocyanin and flavonoids, have a critical role against the UVB radiation, which is considered as the biochemical marker against the UVB radiation, which is rich in rich in the traditional variety, but not in the Swarnaprabha variety. We always focus on the productivity, but with maintain their productivity, and also we focus on the various abiotic stress, like the UVB stress. So if we identify the particular valuable gene of traditional variety and integrate into the high yield variety, so that we develop a transgenic variety which is well sustained in UVB stress environmental condition, which is a very great achievement in agricultural field. And this is my reference. And finally, the good rice is always required for a healthy life, so we always think about it.
Thank you very much. This is very good presentation. How many years you are working with this project? 1 year. Good. Well, now I invite the last speaker, Sanam Kumar Sampal.
Areas adjacent to my wife is Vikram is the groundwater is very important for drinking water in both rural and urban areas, and it can be polluted through natural way. It can be polluted by a natural way or in an anthropogenic way. If we talk about anthropogenic way, then mining is one of the major cause. Then coming to my study area, that is one of the largest mining area in India, and it is located in Angul district of Odisha. And MCL has done mining here, several open mining and underground mining. These are the sampling site of mining. We have taken 30 sampling sites, and from which we have taken samples, and then we collected the groundwater. Then we analyzed the water quality, and then we have analyzed its heavy metals concentration. When we analyzed the water quality index, we found that 14% of the water sample sites are under excellent and good category, while 40% are under very poor category and 33% are poor category, while 13% and 7% are under unfit for consumption category. Then when we analyze the heavy metal concentration, we generally evaluated 3 indices. One of them is heavy metal pollution index, and after analyzing heavy metal pollution index, we found that 33% of the samples which are above the pollution value of 100 show the heavy metal pollution. And second one, when we analyze heavy metal evaluation index, we found that 43% of the samples are higher in the heavy metal containing area. Third one, when we calculated the degree of contamination, we found that 20% has a medium contamination and 80% have low contamination of heavy metal. Then after analyzing heavy metal indexes, we assess the health risk, human health risk that can be divided into either carcinogenic risk or non-carcinogenic risk, which is calculated by 100%. And after analyzing carcinogenic risk, we found that they are within the acceptable limit. So there is no carcinogenic risk as the level of cadmium, nickel, and lead above permissible limit, but there is no carcinogenic risk. Why? There are 20% of sampling site which shows the non-hazardous risk, which can cause diseases like CKD, kidney diseases, and bone illness like this. So it can be concluded that there are heavy metals contamination present in near the mining area of Talcher City. And cadmium is one of the highest contributor. When we calculated the non-hazardous risk, in this 20% of sampling sets, cadmium is one of the major contributor to the non-carcinogenic risk. These are my references. Thank you.
Well done, well in time. Thank you very much. Our next speaker will be Sriman. Take these.
Sriman, I am going to present my topic entitled One Unit treated oral cancer cells for apoptosis. So we all know mitochondria is very dynamic and mitochondrial addition and fusion cycle generally moves in a healthy cell. In cancer, when the cells are treated with cisplatin like drugs, they undergo a hyperfusion state for liver resistance. So we are working on in the how the mechanism of hyperfusion will lead to drug resistance in oral cancer. So first we identified that SIRT1 is a histone class 3 histone deacetylase gene that is downregulated in oral cancer cells, and we found in different types of oral cancer cells, two oral cancer cells have the frequent downregulation, and that is conserved across the cancer cells. Again, we generated one resistance cell, resistance cell that is polyploid giant cancer cells which has a stem cell character and they are resistant, and we found that in the resistant cells also the C1 level is down. Further, we tested how this hyperfusion works. So hyperfusion is a mechanism that happens when the cells, the mitochondrial fission is inhibited, and we check the expression of C1 and its downregulated downstream targeting DNRP1, which is downregulated when the drug is treated. At the meantime, the mitochondrial fission proteins are increased, and when we electrically, genetically introduce the C1, that is in the C1 overexpressed cells, we found the hyperfusion is reduced. As you can see, the mitochondrial branch length generally decreases, and in the treatment type, the mitochondrial branch length is increasing, showing that it is mitochondrial hyperfusion. And further, we quantify this formation by the FIS1 and DRP1 colocalization, two genes that are responsible for mitochondrial fission. Further, we physiologically we challenge how this mitochondrial hyperfusion is occurring. So we first tested that what will be the gene responsible and how the hyperfusion works. We take the mitochondrial DNA nucleotide formation, that is forming a ball-like structure in as in drug-treated conditions, and the ball-like structure holds the mitochondrial DNA to be degraded. While there is drug treatment, the mitochondrial DNA gets degraded by the drug and the cell dies.
But in the microball formation, we localize with the DFAM, which is a mitochondrial DNA transcription factor, and we found that the mitochondrial DNA is entrapped inside the mitobulb, and mitobulb is present. The mitobulb is so consistent with the mitochondrial DNA that when the mitochondrial DNA synthesis inhibitor is given, it is downregulated. It is not degraded. Further, we mark the site where the mitochondrial fission will and we found that there will be no mitochondrial fission when there is mitobulb like formation, and we checked. And while there is a drug treatment, the apoptotic cells generally have the cytochrome C release in the apoptosis, and in the mitobulb, the cytochrome C is stuck. And the mitochondrial DNA is not degraded, and the cytochrome C is so stable that it will not go to the cytoplasm of the cell. So, for that, when the CYP1 is introduced into the cell, there is no mitobulb formation, and the cytochrome C is released here in the 2 and 4. And there is a constriction site is marked, and that signifies that there is no mitobulb formation. The mitochondria will go on fission, and from the fission, the cytochrome C will release from the pores that is created by the DRP1 and wax. So further, we check that the hyperfusion how it works. We check in the mitochondria generally related to ROS. Hence, we check the ROS parameters. First, we check the gene NRF2, which is responsible for ROS regulation. Then we check the cell viability when the CYP1 is introduced. The cell viability in the resistant cells itself is downregulating, and the long-term cytotoxicity is also degraded. And we checked the NRF2 related antioxidant gene systems. Further, we have made one cell line that is primed with the gallic acid, which is a natural activator of CYP1, and found that when the CYP1 is activated by gallic acid, the process of resistance is downregulated. And from the primed cells, when the CYP1 is knocked out, the process is rescuing. Further, in the PGCCs, the resistant cells we introduced, we first saw that the CYP1 is downregulated in the PGCCs. When the PGCCs are introduced with CYP1, the process, the cell viability is going down. That means the PGCCs, which are primarily not resistant to cisplatin, are becoming resistant towards standard. We checked the long-term cytotoxicity by the colonizing assay and checked the apoptosis marker by caspase. So this is the summary of my work. When the cisplatin is treated to the oral cancer cells and its resistant cells, there is CYP1 downregulation. When we ectopically introduced our evaluated CYP1, the cytochrome C release will be there. So the process will be reversed. So I would like to thank my supervisor, Dr. Sunil Kumar Bhutia, for his guidance. I all lab members of CCD. NIT Rourkela and DSP Inspire for providing. So the take-away message I will tell in India, cancer ହେଲେ ଆମେ ମରିବା sure, but ଆମେ antioxidant ସବୁଦିନେ ଖାଉଥିବା ତାକି 5 ବର୍ଷରେ drug resistant ରୁ ମରିବା ସେଇଟା ଜାଗାରେ ଆମେ 15 ବର୍ଷ ବଞ୍ଚିବା. So immunity 50 ବର୍ଷ ଜାଗାରେ 65 ବର୍ଷରେ ମରିବା ଆମେ. Thank you.
Thank you very much. So this is the end of the scholar session and student session. We have a beautiful experience. We once again regret that we could not do justice to our students. Actually, for possibility of time, and we have lost good 25 to 30 minutes due to electricity disturbance. Otherwise, everything would have been fine. Now the last but most important session, last session. I invite Professor Koraput to chair the session and Vikram sir to be the and the first talk will be delivered by Dr. Jeypore. I tell everybody kindly recite your talks.
Sorry, ma'am. I thought William. I tried to finish it as early as possible. Thank you. Uh, okay. I don't have much of a research thing to present here, but anyway, some of my publications I will extract some of the points that will be mostly for the benefit of the students, I think. Uh, Not much time. Sure, I want to listen to young researchers. Anyway, bioactive compounds, I'm sure most of you have heard about this. We have been listening about this right from our college days, way back in 85, 86. Then during our PG days, we had a special paper called phytochemicals. Then we were told that most of the medical needs of humanity is made from plant resources or little bit by animal resources. But nonetheless, natural resources are meeting the demands of our medical aspirations, whether it is in regard to therapeutics, whether with regard to nutraceuticals, or with regard to, say, our food additives, or maybe essence, any which way. But then we have been listening about the plant-based drugs, then botanicals, then bioactive compounds, then nutraceuticals, then finally, these days we are talking about bioactive compounds and more specifically bioactive principles. So I will simply, I have adopted this device. I will simply explain first, then I will rush through the slides so that I can save some time. Anyway, through the bioactive principles, we know many specific biomolecules that has to be extracted. When we need specific bioactive molecules, accordingly, you imagine how much of bio resources you need to consume in order to, say, producing for 1 milligram of curcumin from Holland, you all might be knowing how much of rhizome you need to, okay, rather destroy, I would say, because finally at the end of so much of extraction process, it goes from 10 kilos of curcuma longa rhizome, we hardly get 1 milligram of curcumin. So when our demand for more precision bioactive compounds, say bioactive principles are increasing, on the other hand, we say that all the bioresources that meet our demands are depleting day by day. The depleting and the demand division is growing by age. So in order to meet that high pressure on the product level, what need to be done? We don't have much land left to cultivate all these plants to meet our bioactive compound demands. So we need to get into modern exercises, say whether it is the field or in the lab or in matters of our consumption habits. We need to get into the real issue. As Professor Mahapatra yesterday told that, modern engineering, genetic engineering techniques cannot get to the level of product. Whereas every research now the thrust is on the product, not even the process, the product, the final product you need to deliver. There only the money comes. All our young scientists know that simply genetic engineering for the sake of demonstration of an issue is no more being taken seriously. The funding agencies are giving emphasis on the products. In that sense, we thought no amount of phytochemical analysis, then finally genetic engineering interventions, including crop improvement programs at the molecular level, are hitting to the problem. So we thought, given the land, given the level of bio resources at our hand, how to optimize and predict the yield of these end products of biofuels. So what we did, we mimicked the biological neuron system. Okay, all students must be knowing. Neuron is the unit of information transmission in any biological system. One neuron, it receives the information at the one end, it processes, transmits, and delivers as a product at the other. This function of a biological neuron was taken or mimicked by artificial neural network studies, which compose the part. I think most of you already know about the artificial intelligence regime, the biocomputational regime that is coming into high-end research in biology. What these scientists did, they mimicked the biological neuron by taking all the information at one end, and then they were allowed to be processed and transmitted to the other end. We do not know what is going on in the transmission and processing area. Okay? Now, I will rush through the slides. Simply, I will explain to the students all the information that are fed at the neuron receiving level. It undergoes the processing and transmission and is delivered at the final product. If there is one neuron to neuron, we call it single neuron layer. If there are more than one neurons interconnected, then we are calling it a multi-neuron network. So, scientists, what they did, maybe it is 10 a decade old practice, and many of ODIA scientists are also working, and Kabalpur University, Professor Pradeep Kumar Naik is a master in that. They have a bioinformatics and biotechnology center there. They are only popularizing the artificial neural network model. So, as you know, it is a algorithm model which can recognize the prediction, the pattern prediction, and then optimize and give a kind of forecasting where when to cultivate this particular crop or elite variety so that the yield of the ultimate biofuel principle will be maximized. So end of the day, as you know, it is a prediction model wherein the algorithms, popular algorithms based on the patterns are utilized to devise a model wherein you can definitely specify the location, the elite varieties, what are the data parameters you can feed at the entry level so that it can go through the process and transmission area, and then finally the end product can be predicted properly. That is the basic idea of applying artificial neural network program. These are all algorithm in silico programs you can use to predict optimum yield of bioactive principles instead of straight going to the field or instead of straight damaging all the bioresources physically. You can definitely fit into the model which will ultimately give the highest yield, and then you can go for multi-locational field trial, cultivation, and ultimate extraction and data verification of the end product. That is precisely is the my paper covers. I can okay. As I told you, I will rush through the slides and give you the overall idea about what exactly I am going to tell. That we have this idea of phytochemicals to the bioactive molecule. Now this is the age of biomolecules. Everybody is talking about what is the end product of your research and how commercially viable it would be if it is upscaled to a research. Okay? Okay. Here I want my students to know that earlier everybody used to set aside that okay, don't talk about phytochemicals, plant-based drugs, crude extracts. You come down with the exact bioactive principle molecule so that you can continue with the research. Now, after this carbon program, you must be knowing. Now, no plant extracts is set aside. Nobody just throwing you everything. You are talking very crude level extraction. Come down to the bioactive principle level. But what they are doing, you can go from bottom upwards instead of always coming from top downwards. So the research these days are from you first isolate the bioactive principle first by employing all these biotechnical tools. End of the day, biotechnology can be tools, not the end of any journey. So by using the molecular marker techniques and the SPTLC extraction methods and characterization methods, you can zero in into the bioactive principle as a molecule, and from there all the corollary information you can feed into the artificial neural network and then finally predict the optimum yield model so that whichever is suitable for you, whichever environment is you can definitely go and employ it. So I will rush through, as I told you, these are the methods by you used to extract bioactive principles. Mostly they were told as crude plant extracts. But applying all your wet lab data, you can by GCMS and other chromatographic techniques, you can zero in into your specific biochemical principle. At the same time, you can use the ethnobotanical traditional knowledge system data and phytogeographical data along with your economical details also. You can feed at the entry level of the artificial neural network model a specific, discrete, countable, measurable data so that more precise, more countable, more discrete the data, the final predict will be better comprehended and accepted. Okay. Now, we, apart from all the bioactive compounds for last 10, 15 years, many times we have heard about curcumin being in the patent controversy. So some of our colleagues in SOA University, they started an interesting project they got from TPT, from Odisha, Bihar and Bengal and Andhra Pradesh parts to collect all the thermal germplasm, mostly the elite germplasm, along with some wild cultivars also, and the final extraction of the curcumin content, going to the level of curcumin with its structural and functional principles. Likewise, blastocysts A from Pectopomeriae we just collected, then Shatavari content from what is that? Asparagus, the Sotalu. These plants we have been working for past 5, 6, 7 years, and we got very tremendous result as per predicting, optimizing the final yield of consuming Becoside A as well as Satalu. And it's working wonders. In multi-locational trial, we have seen elite germplasm can be manipulated by their agronomic inputs, phytogeographical inputs, and other wet lab data input. Final prediction can be 100% assured. That is the beauty. As I explained you how it is mimicking the artificial neural network, how it is mimicking a real biological neural. So the basics of artificial neural network I told you, like a neuron, it receives, it processes, it transmits, and it delivers as the final product. We can go further. So many, as I told you, it is all based on the algorithm. Like we have in social media, the likes and dislikes make an algorithm which can be utilized for mapping onto other predictions also. Likewise, this ANN is used to map onto the different available data at the entry level so that the final yield of the specific bioactive principles can be properly predicted. The applications are almost there, right from face recognition systems to predicting the stock market values. Everywhere this INN has been employed, and now only recently, last 1 decade, maybe biological scientists also started using ANN as a prediction model. And that is, as I told you, doing wonders. Okay, there are some of the architectures of ANN we have been using. The first three, perceptron model, the radial basis function model, and multilayer. You can see the first one, this one, the single layer perceptron model, you can see the input data and the hidden layer. Hidden layer means where from the processing and transmission starts. Depending on the number of input layers and the hidden layers and the final output layers, the architecture can be a single layer perceptron model, maybe radial basis function model, or maybe multi-layer perceptron model. In between also there is a feed forward and back propagation methods. Simply the data available, if we are using backwards in the prediction model, that is known as backward propagation model and feed forward model. We try to use both these models, especially the first, second, third, and part of the fifth model. You see, this is the most acceptable model in which we first experimented the curcumin content prediction, wherein we took some input data, basic, as I told you, from phytogeographic, from agronomic, from wet lab data, we all collected at the reception level. We tried to make it as many discrete, specific data possible so that our end product will be that much predictable. So in curcumin, as I told you, this is the data you see from our agronomic to climatic indices we used as a feeding information, and then finally the output was predicted to be 0.5, for example. Now, as I told you, we from different agroclimatic locations we collected the germplasm and we subjected it to basic agronomic as well as biochemical data analysis, including the geographical indices in the shape of contour plot, so that we can finally predict which geographical agroclimatic region will be the most suitable for curcumin, maximizing curcumin yield by using this artificial network model. These are the different types of data, as I told you. The more discrete number of data you use at the feeding level, your prediction will be that much precise. This is for bacterocyte, as I told you. We collected accessions from different areas of Odisha, West Bengal, and beer and partly Andhra Pradesh also, we extracted the bacteria content with the SPTLC and other. You see, the content was scattered plotted in order to have fit into three sets of data. In our artificial neural network, we use a training set of data, the elementary set of data for testing. Then finally, we allow it to be tested in the lab so that the trained set, the tested set, the result can be extrapolated to the final validation data. So that what we predicted at the beginning of your testing can be corroborated with the final predicted data. Okay, I hope you all details cannot be now covered, but then as I told you, what are different types of data you use at the fielding level, you get that much of prediction precision. Okay, now I will rush through the slides. I'm sorry. In the meantime, some statistical significance that testing also you can do by testing the relative sensitivity of all the in-feeding parameters. Okay, likewise, I told you like pesticide and curcumin, you also started with Saxavalin also. Right from the crude expression through SPTLC and study their exhaustion spectra data to corroborate right from the densitogram from the SPTLC 2D graph. We wanted to make it more precise so that Saxavalin content, if the maximum yield is ascertained at one level, along with other parameters, agronomic and other participant geographic parameters, you can more precise biochemical data if you use, then we can not only for Saxavalin 4, we again use an artificial neural network of the multi-layer perceptron method, where you have many number of parameters in the input layer, as many as number possible, corroborated by our training data set as our testing data set and validation data set, so that our hidden layer only at best we have 1, 3, 4. On these hidden layer we don't have much of a control. Hence, the more we make it precise at the entry level, the hidden layer can be the demerits of the hidden layer can be overcome, and you can definitely predict an output. Okay, now Saxavalin, we got the best result in Saxavalin. These are the scatter plot verifications for using. First one is the training data set, the second one is the testing data set, and finally the validation test. The input and output data as given in the artificial neural network can be validated on the scatter plot diagrams. Okay, now we can manipulate with the input data, for example, by manipulating the nitrogen and phosphorus content in the particular agroclimatic zone. You can definitely play with the final output of Saxavalin. These are the overall pictorial representation of the work done specially for Saxavalin. How right from the wide sample collection to final prediction of Saxavalin, this model can be utilized and that can be extrapolated to any medically important bioactive principles, whether they are from plants or microbes or animal sources or they are synthetics or probiotics. From anywhere, this prediction model can be confirmed. Okay, these are some of the publications we had from this work. I think I'm not getting the latest ones. Maybe this one is the Saxavalin one, the earlier one. Okay, SPTLC Saxavalin.1. Anyway, this is a very interesting work. You know, instead of going all the way to wet lab data and only reducing all your time and resources in wet lab data, ultimately you are going to be hit by the product. Everybody, wherever you go, they will ask you, okay, fine, you have done a lot of research, but then what is the ultimate aid and how come it can be commercialized? So in that case, by using dry lab simulation experiment, you can definitely ascertain that, okay, by employing these entry parameters, by modulating these hidden input data, you can definitely predict an highest yield and optimize your yield so that you can suggest which location, which germ plant can be cultivated at which part of the season, so that your yield, ultimate yield of the bioactive principle can be maximized. That is the ultimate objective of this study. Thank you. As I told you, when people are talking about depleting bioresources, whereas our demand for bioproducts are increasing, we don't have much land left, we don't have much biological resources left. In that case, what best we can do is to play with the available techniques so that you can definitely improve your yield to a commercial level without unnecessarily damaging the available bioresources and unnecessarily not fighting for the land. So the land can be utilized for more productive use like your food products or any other immediate, instead of simply throwing money or draining money on a very anticipatory research. You can definitely devote your time and energy and resources in this predictive researches. Thank you very much. I hope I could do justice to the topic. Since I was thinking about somewhere talking something which can relate to the theme of our conference, that is research advantages in biological sciences. So since we are doing a lot of research right from traditional ethnobotany, ethnozoology to the gene level and gene expression research as we have been witnessing for last 2 days, and like Professor Barrier interesting talk about how such gene can be tinkered complementation with the signaling molecule ethylene in order to study the grain filling behavior. Right from today's Dr. Vikas Patnaik's research, how CRISPR gene editing can be helpful in fighting the mutating gene in cornea of human eye. Right from now, so many students and NIT students, especially, they are doing wonderful work under the stewardship of Professor Gujia. I think the next talk will be equally and more interesting from the previous ones from Dr. Gujia, and he will be the icing in the cake. I hope so. Okay. Thank you.
Thank you, Professor. That your very stimulating talk.
Thank you, sir.
But the question is that scientists relies on the real data, not on the modern data. So, I was my third or fourth PhD student. I taught him for 8 years. And the moment he left me, he entered into the JLU and bioinformatics. And he lost in bioinformatics.
Yes, sir.
So that's the pity.
That is the problem, sir.
That's the pity.
Information is, end of the day, is not wisdom.
Right.
But then it can be stored.
Right. Thank you very much.
Thank you, sir.
Next will be Surya, I think. Take over. Thank you.
Hello. Now I request Dr. Jeypore Singh to present his work.
Thank you, Mr. Professor Mahawala, lecture joined very lessons. My esteemed colleague and friend, Mr. and Pragya, from whom we are all proud that you are dust and there is red of the soil. So seeds belong to our area. That's why I am saying dust. Dust means red of the soil. So we are really proud of you. And I congratulate Professor Mahawala and all the team members who are involved in arranging such an interesting seminar that we are in place very eager. I would also like to thank Professor Sahu from Jeypore University, Robert Thesis, Professor Barali sir for coming here and sharing their intellectual thought with the students. I hope that all the students must have benefited. I also congratulate Professor Bhutia that your student is being the convener of the seminar. So the credit goes to Bhutia for producing such a product. I must congratulate Professor and my esteemed colleague we share the same patronage that we are both at taxonomy, Professor Jena, or enlightening us on taxonomy. I am a taxonomy and a proud taxonomy. So like when I start teaching taxonomy, many of my colleagues say this is a boring subject, but I can bet you that it is not at all boring. Anybody who is a good taxonomist definitely will get a job. That I can bet, that I can guarantee. So anybody who is really interested in taxonomy and you spend 4 or 5 years in good research, definitely you will be a scientist or you can be an academician if you are really a taxonomist. And none of subject can guarantee you a job, but taxonomy can guarantee you a job. So what I am going to say is, I am going to focus on integrated taxonomy approach, where Dr. Jena led. Dr. Jena spoke about morpho taxonomy as well as on molecular taxonomy. I would add something to it. I am going to speak some genotype on 2 slides. Also, I am going to speak some genetic morphometric that everybody nowadays from cooking photographs trying to identify that organisms. But that is not a good technique to identify the organism because the method is not yet fully developed. And we are trying to work on that from the photographs, how we can identify the organisms without destroying without collecting any tissue, nothing, with a non-invasive technique. And we have published a good number of articles on that genetic morphometric. That also I am going to elaborate. And if you read my abstract, I have written one line that interdisciplinary approach is very essential. The Chinma sir from Uttar University was focusing on interdisciplinary approach. I will also like add something to interdisciplinary approach, that how interdisciplinary approach is very essential to solve different issues on biology in general and taxonomy in particular, the sustainable utilization of bioresources. So that is the plan. This is what I mentioned, that I am going to focus on interdisciplinary approach. This is not my work, rather I will share my experiences because I started my journey from Zoological Survey of India since 2008, and whatever I have experienced from my field studies, I will share and invite students to work in that way so that we can resolve many issues. See, when we are talking on science, we are neglecting social science people, the humanity people, people from mathematics. That is not a good practice. We should invite them, at least the students, so that we can do interdisciplinary approach. We cannot make the students sitting outside. So we should invite the students across different sphere of life, both from humanity and social science, because they can have bigger roles in biology and other research. So I request the authority of these colleges to take care of that thing, that when we are organizing any seminar, we should also invite the students from Korea because they have a better role. And I will say and I will prove that how they can be a better contributor for biology. Then I will also focus on integrated approach, that what Dr. Jena mentioned, and some case studies from both from published literature as well as from my work. Then I will speak on another topic, that is called invasive alien species, and why that is a growing concern for all of us. So let's start. So I am showing this picture from Sundarban. So I started my journey from Sundarban. I spent more than 90 days inside Sundarban. And this core area of Sundarban is like a heaven. Only lucky people can go, because I was at JSI. I got an opportunity to spend more than 90 days inside Sundarban. Why I am saying this, showing this photograph? There is a core area. See the zigzag pattern of one bush. I am not a plant expert. So, but see, then from here around 1 kilometer you are getting Bangladesh. Suppose you are a taxonomist and you are searching here the animal, and without knowing the political boundary of the state, if you are entering into the Bangladesh, then you know the consequence. So knowing the political boundary is very important. If you are working on any ecosystem and designing a policy, you should know the geology, you should know the geography. Like people are involving in climate change. In Sundarban, there are more than 100 islands, and 2 islands have already been submerged. That's why knowing the natural history is very important. So that's why I am saying knowing the local name from the Odia people or the Bengali people, that is also important. So if you do not know the natural history of any habitat or ecosystem, you cannot design a policy for its better management. So many people without knowing the core area boundary, they are entering into the core area, and the forest department of West Bengal like putting any case, many number of cases in their name. So we should educate people that what is core area, what is buffer area, and what are the precautions we need to take before entering into any buffer area or core area. So another thing, see, we have seen tiger in zoo or captive site. If you see a tiger, real, you cannot sleep for days together. We have seen tiger front to front, and our sleep eliminated for around 5 to 6 days. And there is a cultural belief in Sundarban that once you see a tiger, you have to first him this one baby, which is considered to be the custodian of the Sundarban. So this is the cultural belief that we need to invite from the the local people. That's why I'm saying the people of Odia, people of history should be accommodated in any seminar where you are talking about anything. You cannot talk in isolation. So another thing, as I mentioned, I was a part of in diverse census, and in diverse census, when we are participating, we have to put a declaration that if anything happened to me, I'm the sole responsible. And that is not that you might lose your life living diverse census, and you are the responsible. You have the responsibility. The government will compensate, but you have to put a consignment. So what we used to do is we used to collect the legal material along with the bookmark and the stripes of the tag we studied, and we used to send CMB for DNA analysis. That's why I'm saying interdisciplinary approach is very essential. See this question. The tiger stripe is vertical, whereas in geography it is horizontal. Why it is so? Why this natural question will be solved? People spend lakhs of rupees, the biochemistry people from molecular biology, but they do not solve. But the people from mathematics have solved this. Exactly, I do not know, but you will see this article. This is an article from mathematics people. So without mathematics, without the computer science, cannot resolve biology in a better way. That's why I told you this is very essential. Then maybe that so we need to create a job. Only doing research without job creation has no meaning. When you talk about biodiversity, if you are not providing opportunity, job opportunity to people, then people will not never be involved in biodiversity conservation. So government of West Bengal is doing very good thing that when honey collection, they are involving the local people for honey collections and whatever benefit they are getting, they are trickle down. They are giving money to the local people. So why I am showing this picture? See this. The behavioral ecologists say that the tiger will not see who you are on while attacking. So that's why they put the marks so that the tiger cannot attack from front side and back side. But nowadays, the tiger is adapted to these marks. Then we need to understand how tiger is adapting to this mark. That's why interdisciplinary behavioral approach is very essential. Then another important thing I observed in Sundarban 2009 that one tiger died by the king cobra. Both are predator, not predator in ecosystem. But how this ecosystem food chain is get disturbed and where tiger is forced to eat a king cobra, that we need to understand. And there are publications that why tiger is eating king cobra. We need to understand why this behavior is changing. It is due to climate change that we need to understand. Another particular things I observed in Sundarban that monkey are eating fishes. Why it is happening? We need to think on this like the seniors used to. Another thing is if you go to Bakhali, and you see the red carpet is there on the CP. These are called red crab, and we are in a population survey of this red crab, and you cannot get this red crab. It is there, thousands number, but once you catch it, try to catch this crab, you cannot catch them because they will enter. They will enter into their holes, and each craft is meant to enter into one hole. So we find that we spend more than 72 hours to try to collect a single craft, but it fails. But when you understand their behavior, so when you cover one or two holes, that when you tight down the type, this craft is to enter into the type to collect the zooplankton or phytoplankton, then they again come enter into the respective hole. So when you cover the hole, the craft cannot harm the hole. And another peculiar phenomenon is that one craft will not allow the entry of the other craft. So the understanding the behavior is also important. So that's why I have given the title integrated taxonomy approach. Integrated taxonomy approach means when you talk on macrotaxonomy, you need to have behavioral approach, you need to have neurological approach, you need to have the molecular approach, genetic morphometrics, stereotyping, all the approaches are supplementing to each other. So that's why an interdisciplinary approach as well as an integrated taxonomy approach is important for the biodiversity analysis. Again, see, once you now suppose this crab entering the hole. If you go on digging, you cannot get this crab because the hole is ending somewhere else in parallel to the hole. But you are going digging vertically, you cannot find. And doing this, we found by pouring the plaster of Paris into the crab hole. So the underdynamics of the crab hole is very important as well. So if you go on spending your time horizontally, you cannot get the crab. So that is also important when we are collecting the crab. Another thing, see, the particular thing is each species have a particular sand distribution pattern. So the architect people from mathematics should come up and try to understand why it is so rhythmic. See, the pattern is so rhythmic and it is species specific.
So that residuity need to be understood. Why a small organism is able to distribute this time in such a pattern? Either in biological significance, why it is happening, and what is the mathematics, what is the architect behind it, then you need to understand. Then come to the integrated taxonomy approach. So there are around 2.5 million species, but the estimated number is this is the estimated number, but predicted number is 5 to 100 million species. Each and every minute we are reaching organisms without understanding their value. So let's mention that I should move fast. That when the Earth was like formed around 4.5 million years ago, and the past life appeared around 1.5 million years ago. But today we are saying that it is around 10 million. How it is happening? What is the driving force behind it? We need to understand. So we have already witnessed big mass extinctions, and we are heading towards 6 mass extinctions. And before that, we should discover, because without discovering, we are losing many organisms. Now come to although I should not fight this because many botanists are taking here, but I have taken an interesting example as a case study. This is not my work, rather this is a case study from 2007. This work was carried out in the west coast of North America, and it is a phytoplankton called Pseudorhizia. It releases neurotoxin into the environment. And if you apply the morphotaxonomy, only there are 2 species. But when you apply biological approaches, I mentioned, when you apply all the approaches, there are 8 species. That's why the neurotoxin varies. The chemical composition of neurotoxin is also varied. So then comes to case study number 2. Again, it is not my work. So when you are designing any policy based on only morphological work, you are missing the thing, because you are not giving equal importance to the genetic diversity. Both should come up. Both genetic diversity and morphology should be taken into consideration when you are designing any policy. So let's move past. This species, thinking that in single species, but when they apply the genetics, there are 2 species, and this species is found in Atlantic Ocean, and the distribution was limited to only southern part. But when they apply the genetics, there are 2 clear-cut species. So our policy need to be designed in such a way that both should be combined together. So then it comes to my own work. So this is already my today presented. So I got a project from Odisha DST, because this is the only 3 species. There are 4 species on the family Sibiridae, which are under IUCN list. So we are studying about their genetic diversity in order to have better policy approach to conserve them. So I'm moving fast. I'm not talking on detail. So we are applying. We have applied the haplotype analysis based on Dingo business, and we found 10 haplotypes. So it showed that the Valoga of the population is stable in Odisha. We do not have to worry, because this species is under vulnerable category, and this species is only found in Southeast Asia. So we need to have a concern while designing the policy of this species. So see, another thing, when I talk on job creation, this is near to very Koraput. See, can you say me whether it is same species or 2 species? This is single species. So we are trying to resolve these issues of the species. So see the head values, see the color pattern. But when we did the genetic analysis, they belong to same species. So why this is happening? And we have collected this from Harishankar, we have collected it from Tulwani, we have collected it from Lokhari Wildlife Sanctuary by taking permission from the Marathi along with of India. But we found that all the sequence belong to same species, and the genetic difference is less than 1%. So sometimes genetics also gives you confusing things. So we need to have a we are resolving this. It may be new science. We are trying on that. And since I told on job creation, those found on Koraput should concentrate that if you can artificially interpret these things, then we can create this piece in the aquarium trading. And this is so beautiful, and this can survive. We have seen this species can survive adverse environmental conditions. So we can have artificial breeding on this. So we can have a project on hand. It can create, it can earn money on that. So again, I'm skipping. Let not. Another thing, another integrated approach. I will take another 5 minutes. Then I will. So the vast shape variation is very important. So I talked on molecular approach and morphotaxonomy. Shape variation is. I mentioned it in the beginning that from the photograph you can identify the organism. It is based upon this principle. The linear dimension of the different species may be same, or same species may be different. So, but when at the but the shape is species specific. But the linear dimension length may be different from the same species or same from the different species. So this shape variation analysis is very essential to discriminate between population of the same species. So we apply this and it's a protocol. I am not going in detail. We apply this in the species and we got beautiful results that through severe analysis we got 11 cluster that is can be distinguished from one category to other. So another in this group also we apply we able to conclude that there is differences among these. Another approach we are seeing also we are studying on their studying in their osteology. From the osteology also, from the bone anatomy, bone structure, also we can identify the species. No need to go for DNA for all the cases. So this is what we can differentiate based on the caudal bone. Then come to next is karyotyping. Karyotyping, if you see the pieces, around 30,000 pieces are known, but only 10% of the pieces are known karyotypically. But nowadays nobody is doing karyotype. In Odisha, Koraput University, you want to establish the technique, but now people are not doing. But I have started, but still I am also not doing. So we need to train some students to work on karyotype. So karyotype also gives you good results. Karyotype, you can do a lot of things when you are doing advertisements, artificial breeding. Karyotype also plays a significant role. So I am skipping in the time not there. Another project we are dealing on from the SCRB. So we are studying on the parasitic acid. The parasitic acids they used to eat the entire fish. They used to damage the part of the fish. So they are a really threat to the fishery sector. So we are also focusing on this parasite. The parasite is beautiful because they are damaging the fishery sector. So the parasites are also challenging to identify. Also in photograph of two species, they are so overlapping. So in this case, we can have multidisciplinary approach to solve this case. So I am moving back. Another thing that life cycle is also important. These parasites, they are completing their life cycle in this manner. Male become a female. Interestingly, the male become female, then the female, the juvenile and different stages, then again become male become female. So if you are getting a single stage, then you cannot conclude on anything. So collection of many different mark, not mark, many all the stages is very important when you are designating a species of the parasite. So another thing, the last part that is called invasive alien species is really a concern that if you do not understand the biology of the invasive species, once you are getting an invasive species, then it will hamper your ecosystem badly. So these are the statistics of the invasive species of different countries. So in one of the university campus, we got a species called in 2015 to 18. This ant was prevailing in such a huge number in one of the university campus that it just displaced many native species. So from the behavior of the species, you can identify this ant. So this ant, like you must have heard in OTP that there was news in some village of Puri that ant population is growing and it is really concerned. So this ant behavior you can identify the species. So I am not going in detail. See, another thing is the Australian government spent crores of rupees because it eliminated many native species of crab from Christmas Island. So once this number is growing, it is very difficult to control. So I am not going in detail. We have carried out studies, and one particular thing we have observed that these ants they are mutualistic relationship with many humidora. They used to protect the humidora, and humidora used to feed the ant who is honeybee. So there was a mutualistic relationship. That's why behavior, many approaches required to understand the diversity. So I am not going in detail since many people are studying numbers. So there are invasive studies crazy and disrupting the pumping of products to this world. Another thing that Tamil Nadu government is looking for lantana camera. This invasive species market is growing. I do not know how it is affecting, but it is considered to be invasive species. So thank you. So this is another species that is invasive alien species. Those who are from botany and this is Arconia perspective. You can have a look on this, and it is growing in Gopalpur area in plenty numbers. So how it is going to have impact? Please look after this. So I want to thank the funding agency from this one. I got a project from HCRB, Odisha District, and my collaborator is University of India, and my bread and butter institute. So whatever I have published due to their idea, they come up with the idea, only I try to provide the platform. Without them, I am nothing. So I am thankful to all my students for parting their idea, spending their energy for my development. Thank you very much. So I conclude sustainable today or sustainable tomorrow.
Thank you. There are no questions, please. I invite Dr. Sujit here from NIT Rourkela.
I will tell you quickly as much as possible. Maybe 5 to 10. Because you have I am the last speaker of speaker. So last conference Mundi Hara. Sister many of our members of my students presented. So I will briefly just narrate what we are doing at NIT Rourkela. So we are working on the oral cancer. We are looking for our design indicator. So how we can increase more cell death of the cancer because cancer cell escape approaches. So main aim is how to promote cell death having less side effects. So this is in Bengal and Bihar, Odisha, we have major focus on the oral cancer because we face on and all this. These are statistics I will not tell. So what we are working, I am working in a topic protocol. One is self-continuous eating system. So it is a degradation process. Like we have a discussion, we have to clean our ecosystem. Similarly, the automatically cleaning our system. It's a degradation process. So what in this perspective, what in oral cancer, what is the role of autophagy we are investigating and how we can modulate autophagy for better cancer. So many things we have I cite one example, but it's the benefit of the autophagy. So autophagy are telling is a self-degradation process when cells induce stress. So like we do ekadashi, we do also like our CAR students, they do intermittent fasting to be healthy, more healthy. So this when we do fasting or when we do ekadashi or when we do intermittent fasting, the autophagy is activated and that activates the autophagy leads to degrade the unwanted material from the body and keep our healthy. So our ancients are more intelligent, more scientific than us. So they do ekadashi and all these things and doing autophagy. Some report that they are morning work also doing autophagy. And now I am doing yoga, so I am also interested to see whether yoga induces autophagy to protect the system. So here these are different roles of autophagy, different diseases. We are our focus is on different functions of the autophagy. And this is the various diseases associated with autophagy now. Dysregulation is there. Many things are focusing on cancer. And these are different states of autophagy. I will speak everything. So here in the cancer perspective, so during because autophagy protects the cells, so during cancer initiation, autophagy protects the cancer. Autophagy protects us from the cancer. So because it removes the
cancer is altered cells, so autophagy will try to remove these cells, altered cells from different mechanism. But as cancer is progressing, then our students are we have discussed in the morning, my students only also discussed about that. So in the early, at the last step, we have to inhibit the autophagy. So these are the different functions of the cancer. They have a dual role, both promoting and suppressing. And we are working on a gene, that is tax9. So this is during cancer progression, this gene is deleted. The chemosuppression gene. And so why it's relevant? This work is funded by Alzheimer recently project going on. And we found that this gene is suppressed because of methylation. So I will show some data. So here we did in the animal model that the gene is decreasing. Then we did some in vitro assay. And in oral cancer patient is also decreasing. Then we found interestingly when we treat equally and ethanol, the gene is suppressing also. The gene and the methylation is increasing, whereas the methylation is increasing and the expression is decreasing. So it can correlate to that when you take gutta, pan, and other things. So this gene is particularly focusing on this gene is inactivated. When you take alcohol also, this gene is inactivated because of methylation, and that leads to maybe cancer. We did many more. Then we studied many things. Lot of work we did. So how opportunity is increased, then we studied autophagy, then how it affects the stem cells, everything, how it uses autophagy, and finally, that in vitro assay perform, then we inhibit the survival gene so that there is no cell death, and we at the early stage we can save our life. And we perform in mice model, and this is the summary slide. So when we when we take the carcinogens, including edible pan, vodka, and alcohol, the gene is suppressed, and that leads to cancer development. But what is the function of the gene? This is a tumor suppression gene. It involves in the differentiation and ours. So this is our study. I will conclude here. Thank
Thank you, Dr. Bhutia. This is a nice topic, but there is no time to discuss. So I feel sorry that I intervened. And now the mic is over to the organizers.
Well, now I invite Dr. Jeypore to come over here, and I invite Professor Sahu to felicitate Dr. Patnaik. Thank you.
लाइट दे दो ना इसको लाइट दिया हाँ लाइट लाइट अरे दिया हाँ
Professor Patnaik, thank you, Professor Rao. Now I invite Professor Jeypore to come over here. May I invite Professor Career Islam to come over. Now I invite this staff to come over here and present their staff. Please. Thank you very much, everyone. Now it is tea time. 5 minutes immediately after tea, we are going to have a valedictory session. And we are going to. Thank you very much for everyone's cooperation. It is still time. All speakers are covered. Please don't go outside. He will be stopped at your right at your place now.